Hello, my name is Bing Han, working at the National Center for Gene Research, Chinese Academy of Sciences in Shanghai, in China. I was doing the PhD study in the Sainsbury Laboratory, Joint Center in Norwich, in UK. I still remember the historical moment that symbolized the end of the Cold War. When I was a child, I wanted to be an engineer. This might be I was growing up at a factory environment. My mother used to work as a consultant in a factory. But since I took my study for biological sciences, I have been fully enjoying research in plant and microbial genetics. By using genomic approach, my research breaks the wall that blocks the link between phenotypes and genotypes. That is, my research unlocked the puzzles of the rice domestication and the genetic mechanism of the rice hybrid vigor. The challenge is that most of the plant phenotype and agronomic traits are controlled by multiple genetic factors and environmental conditions. It is also called complex trait. So my solution is to collect big and genetically diverse rice samples together with the newly developed genomic tool to address the genetic basis of complex trait, hybrid vigor, and to unlock the origin of domesticated rice. My research has been focused on the rice genomic sequencing and genotyping efforts that have contributed to understanding of quantitative trait, domestication, and the genetic basis of hybrid vigor in rice. Firstly, I developed new genomic approaches for tapping diverse rice germplasm to create enhanced capacity to perform high-resolution genotyping and genome-wide association study of complex traits. So this study was the first case and a milestone work for genome-wide association study in crops. Secondly, by constructing the comprehensive rice genome variation map of geographic diverse excision, including white rice and cultivated rice, and searching for signatures of artificial selection, my research unlocked that the Asian cultivated rice has undergone single origin for domestication in southern China. Thirdly, plant hybrid vigor means that the offspring heterozygous plants performed better than their parental inbreeding lines. However, the genetic mechanism of heterosis in crops has long been a puzzle, despite the fact that the hybrid vigor has been discovered more than a century ago. By using genomic approach, I performed the largest scale genomic mapping for grain yield related trait and heterotic effect in thousands of hybrid variety, and characterized the genomic architecture of heterosis for yielding tree. With the success to review the mechanism behind its biological and agronomic important phenomena. So I identified that the accumulation of the numerous real superior alleles with positive dominance is an important contributor to the heterotic phenomena. This result informed on the genomic architecture of heterosis for yield trait in rice and a new strategy for future crop improvement that is possible to achieve a high level of grain production without the need to construct a hybrid by crossing inbreed parental lines in rice. I believe that my rice genomic research has impact for generations to come and yielding enormous insights into basic plant biology. So the genotype and the GWAS demonstrated that the quantitative trait gene can be effectively identified on the basis of the genome-wide association study. The rice domestication study provided an important resource for rice breeding and an effective genomic approach for crop domestication study. And the rice hybrid heterosis study clearly demonstrated the genomic architecture of heterosis for yield traits in rice and informed a new strategy for future crop improvement. So how different heterotic genes contribute quantitatively to the heterosis phenomenon in rice, and what is the genetic or genomic principle for quantitative trait? These are the remain unanswered questions. I will continue to address these challenging questions by using an integrated genomic approach. So which part of my research keeps me lying awake at night? To quantitatively detect the effect of the heterotic gene contributing to yield trait always keeps me lying awake at night. This is indeed a novel and an exciting research project, and is also a big challenge research project. My best friend Tao Wang, a computer expert who has witnessed most of my research activity since I came back to China from UK, he believes that my research works are among the most notable epoch-making achievement in rice genomic research since the elucidation of the whole genome sequencing.